Hello, I'm Yuri Inoue, first grade PhD student, medical science, Takahashi Laboratory. Takahashi Laboratory have around 40 members. Let's first do short lab tour. Our lab is divided into three main groups and other small groups, namely Space Mouse Kibo and Sugar Chain Group, MAF A and CMAF Group, MAF B and Imaging Group. Now, Takuto Hayashi, second grade master student in Frontier Science, part of Space Mouse Research, will introduce about Space Mouse Research.
We aim to identify genes related to new skeletal muscle atrophy by using a mouse housed in space. Mice were housed in a microgravity environment for 35 days in the CBEF in the Kibo experiment building of the ISS. We used the mice after returning to the Earth to analyze various skeletal muscle changes between microgravity and 1Z. We select genes with different expression between microgravity and 1Z and identify muscle atrophy-related genes by various methods. The methods include in vitro analysis using cell lines derived from skeletal muscle and phenotypic analysis related to skeletal muscle function using genetically modified mice.
Now, Riku Suzuki, third grade student of PhD in human biology, will brief about sugar chain research in laboratory.
The aim of our research is to elucidate the biological function of sugar chain. This picture shows the surface of cell. Sugar chain is bound to proteins, lipids, and other small molecules, and it contributes to important physiological functions, for example, regulation of signal transduction, cell proliferation, development, and differentiation of cells. Our group especially is focusing on mucin-type sugar chain, which is bound to protein. In order to elucidate physiological function of mucin-type sugar chain, we are using bias conditional knockout mice. For example, mice with systemic loss of mucin-type sugar chain show pancreatitis, glomerular injury, atrophy of adipocyte. We also have tissue-specific knockout mice and are trying to clarify what kind of sugar chain are important in each tissue. Join us and let's do fantastic glycation research.
Now, Masaharu Yoshihara, a first grade student of humanics, will brief about large MAF and diabetic model mice.
MAF is a transcription factors which consists of several proteins including MAF-A and MAF-B. MAF-A and MAF-B has already been reported to associate with human diseases such as insulinoma. Our laboratory has already established several kinds of MAF-A and MAF-B transgenic mice show that MAF-A and MAF-B cooperatively works in a beta cell which produces insulin. Our aim of study is to establish novel therapeutics which is against diabetes mellitus.
Now, Mitsunori Fujino, first grade student of PhD in human biology, will tell about CMAF research.
The transcription factor CMAF belongs to the large MAF family. CMAF share a basic region and leucine zipper domain. In various tissues, the function of CMAF have been widely studied and reported have suggested that CMAF play a critical role in cell fate identification and cellular differentiation. There are examples of human mutation of CMAF. In 2002, it was reported that point mutation of CMAF was the cause of human congenital cataract and iris globoma. In addition, CMAF knockout embryo mice showed lens fiber cell differentiation and lens formation due to the regulation of crystalline gene expression during the differentiation of the primary posterior lens fiber cells. These results indicate that CMAF is important to lens maintenance. However, a postnatal function of CMAF have remained largely unknown because CMAF null mice exhibit embryonic lethality. Therefore, we generated conditional knockout mice and we are checking the phenotype in CMAF conditional knockout mice.
Dr. Kaushalya Kautunga, postdoctorate in our laboratory, who reported novel IRFP system for in vivo mouse atherosclerosis imaging, will tell about her research.
Hello, I'm Kaushalya Kulatunga, a postdoctoral researcher from Sri Lanka. I'm a member of MAFB group of Takahashi Lab. Our subgroup is interested in development of in vivo imaging systems for imaging various diseases in mouse model. We have successfully developed a new in vivo atherosclerosis imaging system in mouse, which have published in scientific reports in 2018. In brief, we have used IRFP fluorescence as the imaging probe to image atherosclerosis for the first time. IRFP transgenic macrophages accumulation in atherosclerosis plaque was imaged by IV system. Our imaging system has the advantage of imaging the atherosclerosis development in the same mice as the time course data and could image 5 mice at the same time effectively. We are using our imaging system to screen potential treatments for atherosclerosis and also working on development of new in vivo imaging systems for other diseases.
At last, Manoj Kumar Yadav, 5th grade student of PhD in human biology, will shortly brief about MAFB research in macrophage.
Myself, Manoj Kumar Yadav, and I am PhD 5th grade student in human biology. I joined this laboratory in 2015. My work is mostly focused on MAFB and macrophages related study. MAFB, which is also known as musculo-neurotic fibrosarcoma oncogene B and is a B-zip transcription factor. We used the macrophage specific deletion of MAFB in mice and identified the role of MAFB in macrophages, the downstream of MAFB, which is AIM, apoptosis inhibitor factor, and regulates metabolism and atherosclerosis condition. We also reported another important downstream of MAFB, that is C1q, and its regulation in macrophages affect apoptotic cell clearance. More recently, we have published that MAFB is useful marker for tumor-associated macrophages for both human and mice. Now we are focused on studying the role of MAFB in macrophages in relation to metabolism, thermogenesis, wound healing, and tumor development.
In Takahashi Laboratory, we do research and enjoy the taste of science. We do a lot of group party, including birthday party together in our lab. I thank you to all Takahashi Lab members who helped to make this video, and I also thank you for all support from our all lab members. At last, I thank you to Professor Takahashi for his great support in his laboratory to all members and providing opportunity for all of us.